Well, we're off to Baltimore now. The eastern seaboard of the United States might not be the first place you'd go in search of coral, but in the labs of the Carnegie Institution for Science, there are tanks of the beautiful, colourful creatures. Watching over them are scientists interested in how coral deal with changes in the marine environment. An issue even more pressing this week as new figures report an 89% crash in the number of new corals on the Great Barrier Reef following the mass bleaching of a couple of years ago. Well, over there in Baltimore, Roland Pease was in the lab recently with researcher Yishan Jing.
And this room, we started over a year and a half ago, and the idea is to try to grow different corals to find which one grow fastest and easiest in terms of to maintain.
Two large fish tanks filled the lab, one with hard stony corals, the other looked like it contained seaweed, but these were also corals, soft-bodied flowers of flesh.
So here we have 4 different soft coral species, and the ones that's waving, those 2 are called zinnia, pulsing zinnia. They're pulsing their polyps. Each individual polyp have 8 tentacles, and these tentacles pulse rhythmically.
I mean, they look a bit like trees, so they've got a big, thick stem at the bottom,
and
then they branch out. Is that one organism?
So it's one colony. One organism is actually one polyp, which comprises of the stalk and the tentacle. They are colonial organisms. They are interconnected. Each individual is right next to the next individual.
So the same
is
these
over
ones.
here.
These look like petals floating and wafting.
Right. So again, you can see this is clear to see in this pink pulsing zinnia, where you can see individual stalk, and individuals have 8 tentacles. That's one individual. You see that?
So
you can count them.
gorgeous watching those.
Yeah, it's really pretty.
Do you ever come down here when you're stressed?
Yes.
But they're not here for the relaxation, they're here for the science. Yishan is by no means the first to bring corals into a science lab, but she does believe she can bring to coral science molecular techniques necessary to advance the biology.
There's definitely a huge field and also a fairly large literature that has really done a great job at describing corals from the mostly physiological aspects, how corals behave and why they are bleached and why they die after bleaching events. So really we want to understand the molecular mechanism, what molecule, what protein, and what gene is controlling the basic biology of coral.
Particularly the bleaching, a reversal of a deep symbiosis that makes these immobile animals rather more plant-like.
One major basic biology of the coral is that they are able to take up algae called dinoflagellates, meaning